So first we should know class sorry for that. So the bacterial examination of the water, first we should know about the water collection. So how it should be collected. So always remember, the water for any testing, for treating, or for it should be collected in a heat sterilized and screw cap bottles. So this is very important because you should not contaminate the bottle itself before collection. And it should be remembered that whatever the coliforms we are talking, those are the saprophytes. They remain they will be everywhere. So even a small contamination before the collection will give the wrong results. So always you should take the heat sterilized screw cap bottles. So at least 200 ml should be the capacity. So when you are collecting from the tap water, so how you should collect? So the water should be collected only after running the tap for minimum of 2 to 3 minutes. Let it run for 2 to 3 minutes, then collect the water. If at all it is from the streams or the lake, if it is a stagnant water, then the bottle should be opened only after immersing at least at a depth of 30 centimeters. And if it is a well, always try some heavy weight object like the stones or something to the bottle, and then just drop it into the well. And transport, it should be transported as early as possible, at least within the 6 hours of collection. It should not be delayed. Then once it reaches the lab, then it will be gone for the testing. So the first method is plate method or the plate count. Here what we do is, here we check the number of colonies formed in the 4 plate cultures of water samples on the nutrient agar that is incubated aerobically at 37 degrees centigrade for 1 to 2 days. So just what we do is, we take the water and we put it on the we just drop it on the plate. So that is what we call it as a 4 plate method, and we just incubate it and see after 1 to 2 days. We usually incubate it at 2 degrees and 2 different temperatures, 22 degrees centigrade and 37 degrees centigrade. So 22 degrees centigrade indicates the quantity of decomposing organic matter, and increase in the plate count indicates water is contaminated with the parasitic or the pathogenic organisms. So here at 37 degrees centigrade, it indicates some defect in the filter beds to be corrected immediately, and contamination is of animal or human origin. So always it should be incubated at 2 different temperatures. So apart from that, we have some other tests called the presumptive coliform count test, or what we call it as a multiple tube test. So here in the multiple tube test, what we do is, the media what we use here is the McConkey purple broth, or what we call it as a double strength and the single strength McConkey. Okay, so this is the media what we use, and we just inoculate the water into this media. As we know, whenever there is any growth of E. coli in the media, what the color it is going to turn is it is a lactose fermenter, so it turns into the yellow color, and it produces gas, minimum little amount of gas. So when you put a Derham's tube into this test tubes, you can see the collection of the gas in the Derham's tube. So always, so you can use a McConkey purple broth. So here what we do is, I'll just show you a picture. So this is how we collect the we do the testing. So this is the water what we have been collected, that is the 200 ml of water. Then what we do is, we are going to put in the different strengths of the McConkey. So first we are going to take 150 ml bottle. So 150 ml of water and 1 bottle of double strength media. So 50 ml is added to the 50 ml of broth. Then we are going to take the 10 ml of water in 5 tubes with the 10 ml of media, and 1 ml of water each added to the 5 tubes with 5 ml of media. So first we are going to take 5 tubes, 10 ml of the media and 5 ml of the media. Then we are going to add 5 ml and 1 ml respectively. So it is that first we are going to take the 50 ml water, then we are going to take the 10 ml water, then we are going to take the 1 ml water. So this is how we are going to put water in the different volumes of the McConkey broth. So then we are going to incubate it. So once we incubate it at 37 degrees centigrade for 48 hours, then we are going to look at the tubes whether there is any change in the color or not. As I said earlier, because they are the lactose fermenters, so if at all any coliforms are present like E. coli, Klebsiella, Citrobacter, any bacteria which is present, this McConkey broth will turn into yellow color. And because we have put the Durham's tube, if at all there is any gas production, that will be collected in the Durham's tube. So both we have to look for the color change and the gas production. So these two things will be seen. And after this, what we are going to do is these things depending upon depending upon and how many tubes have been turned into yellow color. See, for example, if the 50 ml we are taking 150 ml, 5 10 ml and 5 1 ml. So we are going to check how many tubes have been turned into yellow color. So for example, we are if the entire 50 ml bottle is turned into yellow, we are giving the score as 1. Then among the 5 10 ml tubes, if 3 tubes have turned, then we are giving the number as 3. Then in 1 ml, if 2 tubes have turned, we are giving the number as 2. So 3 plus 2 plus 1, it comes around the 6. So this 6 score will be equivalent to the most probable number. The most probable number will be given. It is given in a chart that is called the McCready's probability table. So we will see that table, and we are going to give it as the presumptive coliform count per 100 ml, whether it is 0 or 1 to 3, 4 to 10 or more than 10. If at all 0, that means there is no color change either in the 50 ml bottle or in the 5 10 ml bottle or in the 5 1 ml bottle. That means there is no change. That means there is no bacteria. So everything is 0. So we are going to give the report as excellent. So the water is excellent. So they can be given for drinking purpose or cooking purpose or whatever. So if at all the NPN number comes between 1 to 3, that is either it is 1, 2 or 3, then we give it as satisfactory, but we ask them to keep checking regularly. If it is 4 to 10, then we call it as suspicious and we ask them to treat it. And if it is more than 10, we give it as unsatisfactory and we tell them it has to be thoroughly treated. So this is one method, that is just by checking the color change and acid production. The second method is what we do is once there is any change in the color and gas production, we are going to put it on the plates. So we are going to put them on the plate and again incubate them, and we are going to identify the bacteria. If at all there is any E. coli, then it should be reported immediately because it indicates the fecal contamination. So it should be informed immediately. So the next one is so this method is called the presumptive coliform count. That is just by the change in the color and the acid production, we are just saying it as a we have just giving the report as a satisfactory, excellent or unsatisfactory. So that is the presumptive coliform count. And whereas this is the differential coliform count, here what we do is it is used to determine whether the coliform bacilli detected in the presumptive test are Escherichia coli or not. So we are more worried whether it is the Escherichia coli, that is the colon Escherichia coli, that means it is the fecal contamination or not. So here how I told you, so once there is any acid production or the gas production, what we do is we'll take that particular sample and we again incubate, we again streak it on the plates, and we see, identify the organism, whether it is a E. coli or not. So that is what we call it as a differential coliform test, or we also call it as the Test-Eijkman test. So we have the other things apart from the differential coliform test. We have the membrane filtration test. As the name suggests here, the measured volume of the water, it is filtered through the membrane with the pore size of 22 micrometers. Then this membrane filter is with the retained bacteria is placed on a suitable media. The colonies will develop within 24 to 48 hours. Then we are going to identify it. Then we are going to give the report. It is just that you are making the water to pass through a membrane filter. Then, apart from this identification of the E. coli, Klebsiella, or coliforms, we have something called detection of the fecal streptococci. So, this presence of Enterococcus faecalis suggests it suggests the recent fecal contamination. So, here what we do, the bottles positive for the growth of presumptive coliform are test are subcultured to the tubes containing 5 ml of glucose azide broth. See, it's like first you are going to do the presumptive coliform test, and again you are taking the same thing and we are going to put it in the glucose azide broth and incubate at 45 degrees centigrade. And this Enterococcus faecalis is indicated by the presence of acid within 48 hours, and it is confirmed by again plating on the McConkey. This is how we detect the fecal streptococci. Then the other most important pathogen is Clostridium perfringens. So the present it is usually present in the water for longer duration. So even if if you find Clostridium perfringens, it doesn't indicate any recent infection. But if at all you find both Clostridium perfringens and E. coli at the same time, then it indicates it is the recent infection, because always the E. coli doesn't remain in the water for the longer periods. So it cannot survive in it usually do not survive in the water for the longer period. So whenever you identify E. coli in the water samples, that means it is the recent infection. Anything Clostridium perfringens, it doesn't give you any clue whether it is a recent or not, because it lives a very longer duration. It remains in the water for a longer duration. It indicates fecal contamination, but it is not recent. So here what we are going to do, here we are taking the varying quantities of water samples, and it is incubated in the litmus milk media at 37 degrees centigrade for 5 days. Usually we get the stormy clot formation, that how you have read in the Clostridium perfringens Clostridium chapter. It gives the stormy clot formation. It indicates the Clostridium perfringens. So apart from this, we can do testing for other pathogenic bacteria if at all required, just typhoid bacilli, that is Salmonella typhi, cholera, Vibrio cholera. It is just that you have to get the water and you have to put it in the selective media. You all know that Salmonella will have they have their selective media, and Vibrio also has the selective media. So you just put it in the selective medias and just wait for the growth. If at all there is any growth, then you can give it as a positive for those bacteria. And viruses, enterovirus and cytopathogenic viruses may be isolated from the drinking water. And as far as the protozoa are concerned, again, Entamoeba histolytica, Giardia, and Balantidium coli may contaminate the drinking water. So this is about the water contamination. So all you should know in the water testing is how you are going to collect the water samples. So in different areas, what are the waterborne pathogens and how you are going to test it. As far as the testing is concerned, you have three methods. One is the presumptive coliform counting method, differential coliform counting method, and finally it is the membrane filtration method. Now moving on to the bacteriology of air. So we all know what are the airborne infections. It is transmission of the infection produced by the respiratory droplets less than 5 micrometers, and it is transmission of the infection produced by respiratory droplets which is more than 5 it should be more than 5 micrometer in size. So always when we are checking the air quality, it should be it is both, we are talking about both the outdoor air and the indoor air qualities. So outdoor air, it depends on many factors. As I told, as we are discussing, it depends upon the density and the human population, nature of the soil, density of the vegetation, and other climatic conditions. Whereas what we are much worried about is indoor air pollution. That is how the air environment is there, how the environment is in our OTs. like how it is in our wards. Then the bacteria which are distributed through the gross droplets, droplet nuclei from the nose, mouth, and dust particles. So why this is very important? We have to know this because it helps in taking the controlling measures so that the respiratory pathogens won't spread within the hospital from one patient to the another patient. So, usually the droplets and the droplet nucleus are expelled in varying numbers during coughing, sneezing, and talking. This is what we know. And most important ones which we are worried are within the indoor settings, usually are the hemolytic streptococci and the tubercle bacilli, diphtheria bacilli, and the staphylococcus. These are the pathogens which we are very much worried about. So, as I told, droplet transmission, it is more than 5 micrometers. Airborne is less than 5 micrometers, and these are the bacteria you should always remember. And this is what we should check for, or we should be treating our air inside the wards and the operation theaters. So what are the methods how we check the quality of air? So the first method is the sedimentation method. This is the crude method or the old method what we are using, or what we also called as settle plate method. This is what this is the method where the petri dishes containing the agar media of known surface area are left open for 30 minutes to 1 hour. Then these plates are incubated at 37 degrees centigrade for 24 hours. So this is what we do it. We just take the petri dishes, that is the agar plates, and we go and we keep it in the we keep open in the any indoor settings like your operation theaters or the wards or any ICUs or anything. We just keep it in open in the keep it open, and we just leave it for 38 minutes to 1 hour. Then we just get back those plates and we incubate it 37 to 47 37 degrees centigrade for 24 hours. The whole point is, so large bacteria carrying the dust particles settle onto the medium. So whenever you are this settling, that is why we call it as a settle plate method. Then we are going to count the number of colonies formed on the plate. And this number of colonies that are formed indicates the number of settled particles containing the bacteria. So this is how we are going to detect or demonstrate whether the bacteria is present in the air or not. So we are going to take the petri dishes. We are just keeping it open in the indoor environment for 30 minutes to 1 hour. We are just getting it back, keep it in the incubator, then checking whether the bacteria is there on that plate or not. So usually we use the blood agar plates, and also we can use the salt extract agars. The second method is the slit sampler method. Here what we are doing, instead of we are just leaving it to the environment, what we do is it is the most convenient for counting the number of bacteria carrying particles suspended in the unit volume of air. That means you are counting how many bacteria or the how many suspended particles are there in unit volume of air. Volume of air. So here there is an equipment. One equipment will be there that will be taken, and it sucks a particular volume of the air, and it throws that volume of the air into the petri dish what you have put at the back of the machine. Okay, sorry, I could not get the picture of that. So it is something like a vacuum cleaner. So it will just suck the air, and while it is putting, if you can remember, the every vacuum cleaner will have a dustbin kinds. So at that particular point, you'll keep the petri dish. So this volume of air, when it is passing through the petri dish, if at all there is any bacteria, it will get stuck to the petri dishes with the agar, and it will start. Once you take it or take out the plate and keep it in the incubator, it will start multiplying. So that is what we call it as a slit sampler method. Usually it has, as I told you, it has three parts. One is the suction pump, and it has a petri dish plate holder, and the outer surface of the slit is usually 0.3 mm width and 27.5 mm length, and it has a capacity to suck the particular volume of air. So when we can give this particular air, is the quality of air is good, or if it has the it has to be treated. So that what we call it as the acceptable limits of air pollution. See, the upper limits of bacterial count in air in the areas will be usually in case of offices or homes, it is 50 per cubic feet in, then 10 per cubic feet in case of operation theaters, and 1 per cubic feet in operation theater for neurosurgery. So this is the acceptable levels of air pollution. So apart from this, we do have some other gross methods for testing the air. So, or testing the quality of air, it is something like checking for the number of exchanges, air exchanges per hour. If at all there are any 20 air exchanges per hour in any OT, or 30 air exchanges per hour, we call it as APH levels. So whenever it is so many air exchanges per hour, we assume that the air is in a good quality. So but again, those are the crude methods. So if at all we have to know, we have to go for this particular methods only. That is two things. One is the settle plate method, other one is the slit sampler method. So that is about air testing of the air. Now moving on to the bacteriology of milk. So we all know this milk can be contaminated at many levels, either from the animal origins or from handling the uh, uh, the milk can be contain bacteria. It can be derived from any source, that is either from the animals, that is from the fecal contamination, or through the infected animal skin, or etc., or from the hands of the milk handlers, or from the environment. So these are the sources of contamination of the milk. So this can be again moving on to the pathogens. What are the what are the milk-borne diseases? What we can see, again, the primary importance will be, we all know the most important one, Mycobacterium bovis, which causes tuberculosis. Then we have the most important one, that is brucellosis. Then Coxiella burnetii. Then apart from that, we have very less important ones, that is cowpox, hand, foot, mouth disease, anthrax, leptospirosis. So these are the things. And there are some diseases which are primarily of human origin and that can be transmitted, that is like typhoid, paratyphoid during the transport of the milk, cholera, shigellosis, etc. And
Excuse me, ma'am. There is voice, ma'am.
oh, sorry about that. uh, is it from the bacteriology of milk? Would anybody respond to me?
Yes, ma'am.
It is from the bacteriology of milk, is it?
Yes, ma'am.
Okay, fine. Yeah. So when we are talking about the bacteriology of milk, so always remember what are the different sources of contamination of the milk. So when we are talking about the contamination, so it can be either from the animals directly, like by the fecal contamination, or through their infected skin, or anything. And second thing is from the hands of the milk handlers. So either you can get the infection from there, from them. And the third one is always the environment. So these are the three sources. So when we are talking about these bacteria or the pathogens, usually we are worried about the diseases that are primarily animal origin and diseases that are usually human origin. If you can see this, so usually of human origin, when we are talking about the human origin, it is the tuberculosis, brucellosis, and we know it. In as far as the tuberculosis is concerned, it is not the regular Mycobacterium tuberculosis. It is always the Mycobacterium bovis and brucellosis, of course. And apart from that, the less important ones will be cowpox, hand, foot, mouth disease, then anthrax. So all these things we are worried about. When we are talking about the infection or the bacteria that are infections that are originated from the human origin, again we have to think about handling. So either it can be cholera, shigellosis, typhoid, and the paratyphoid, and some non-diarrheal diseases like diphtheria, tuberculosis, and other enteroviral infections. So during the handling, if the hand hygiene is not properly mentioned, is not properly followed, so usually these are the diseases which will be spread. So once we got to know this, so we have to know what are the different methods of sterilization of the milk. So always I mean, we already learned in our earlier classes this how we are going to sterilize it. So the first and foremost one is this, the heating of the milk, that is like thermicized milk. We are just going to heat it up for 15 seconds at 57 to 68 degrees centigrade. Apart from that, you have the most famous method called the pasteurization method. I think you all remember the pasteurization method. It has two things, that is, one is the Holder's method, the other one is the flash method. In the flash method, you are going to raise the temperature to almost 72 degrees centigrade, and you just keep it for 2 to 3 seconds and again cool down. And in case of Holder's method, again you are going to hold it for a few seconds. So what happens is, it has been seen that the bacterial spores of Coxiella or Mycobacterium and some other fungal toxins are not being destroyed effectively by pasteurization. It has been found that pasteurization does not kill a few of the toxins and the spores. There is something called the ultra heat treated milk. So here what they do is, the milk is exposed to very high temperatures of 135 degrees centigrade for just 1 second. So these are the methods for sterilization of the milk. So we should know how, if at all, these methods of sterilization have been acceptable, or is it a correct method, or whatever they are doing, they have done is have killed the bacteria or not. See, in case of this helps out in case of dairy farms or any big industries, like for example, take any milk product factories. So they'll treat it. They say that they have doing the pasteurization process, but at the end of the day, you should always check whether the process is proper or not, whether it has killed the bacteria or not, because again, it is of public health interest. The milk will be distributed to the many villages or the towns, so you should always check for whether it has to go through the bacteriological examination. So to check this, we have different methods to check whether the milk is properly sterilized or not. The first one method is the viable count method. So here what we are going to do, here we are just performing the plate counts with the serial dilution of the milk sample. Just take the milk sample and dilute it in the serial dilutions, and you just put it on the plate. So this is one way of identifying. If at all there is any growth, then you can say it is not acceptable. Then test for coliform bacteria. Again, same thing. Take the Mekong agar, take the serial dilutions of the milk, and just put it in the plate, and you just see for the growth. Apart from that, we have some other tests called the methylene blue reduction test. So this is the what they say is the it is the more an economical one, and the more than the viable count method, and it almost gives the instant results. You need not wait for the bacteria to grow. So it usually depends, the what is the principle of this is methylene blue reduction test. It depends on the reduction of methylene blue by bacteria in the milk. For example, if there is any bacteria in the milk, the methylene blue will be reduced. It is as simple as that. So what you have supposed to do, you take the milk, and put the methylene blue in that. If at all there is any bacteria, there will be color change is from blue to pink or colorless. That means the bacteria is present in the milk. That means the milk is not acceptable. It is an immediate test, and it is can be done, and the results will be given much earlier than the viable count method. So it is as simple as methylene. If at all methylene blue is, whenever there is any, if there is any bacteria, the methylene blue will be reduced. So the blue color will be converted into pink color or colorless. So that means there is a bacteria in the milk. There is bacteria in the milk. So that milk is not acceptable. So this is one method. Second method is phosphatase test. So this phosphatase test, it is usually we use it for the checking the effectiveness of pasteurization. As I already told you, it has been seen that some bacterial spores and fungal toxins are not killed by this pasteurization method. So to see the effectiveness of pasteurization, we do the phosphatase test. So in case of here, what is the principle is, effective pasteurization should inactivate the enzyme alkaline phosphatase, which is normally present in the milk. So this phosphatase enzyme breaks down phenyl disodium phosphate to release phenol. So it is that residual phosphatase activity indicates that pasteurization is inadequate. So again, I'm going to repeat it. So if at all there is any, if the pasteurization is perfect, then it should inactivate the enzyme alkaline phosphatase. So if at all there is any phosphatase activity even after pasteurization, then that particular milk is not acceptable. That means the pasteurization is not complete. So that is how this test is useful. It is going to check for the presence of phosphatase enzyme in the pasteurized milk. So how we are going to detect this phosphatase enzyme? It is just by adding the buffer containing the phenyl disodium phosphate to milk. So just imagine like a can of milk has been claimed that has been pasteurized, and it has been got to you for testing. So what you have to do is, you have to just put the buffer containing the phenyl disodium phosphate to the milk. A color development buffer is added, and the tube is incubated at water in 37 degrees centigrade for 2 hours. So if there is any color change in the media, then it means the phosphatase enzyme is still present. So if there is any no color change, that means phosphatase enzyme is not present. So always, if at all there is any effective pasteurization, there should be no color change of the milk. Then that means that milk is acceptable. So this is the phosphatase test. So then the final one, it is the turbidity test. It is usually used to check the if the in case of boiled milk, all the heat coagulable proteins are precipitated and it does not become turbid. So here what we are doing is, the milk is heated first up to 100 degrees centigrade for 5 minutes, and then ammonium sulfate is added. So if once you add the ammonium sulfate, if there is any absence of turbidity, it indicates that milk has been boiled completely for 100 degrees centigrade. That means there should be no turbidity even if you add the ammonium sulfate. So this is the turbidity test. So one is the methylene blue reduction test. You are adding the methylene blue. If it is turning into the pink or colorless, then that milk is not acceptable. Then phosphatase test, you are adding the buffer containing the phenyl disodium phosphate. If it is changing into the pink color, again it is not acceptable. Turbidity test, you are adding ammonium phosphate, and if it is becoming turbid, then it is not acceptable. So these are the three tests what we are going to use for bacteriological examination of milk. So finally, we are moving into the bacteriology of food. This is the most famous one, or the most common one what we see, because most of the times we see that people are suffering from the food poisoning, and usually it is an outbreak. It is not just one person or one will be suffering. So because the source, depending upon the source, it causes an outbreak. So there will be, they pose a significant public health problem causing morbidity and mortality. So and we all know what are the agents which causes the food poisoning. Say outbreak of foodborne infections when two or more persons experience a similar illness after the ingestion of the same food, and epidemiological analysis, it implicates the food as a source of illness. So usually, what are the sources of food contamination? Either it will be feces, sewage, flies, hands of the individuals, air, and the domestic animals and the pets. So these are the different sources. So for testing the food, whenever there is an outbreak, for example, if from a hotel there is an case of there are some 100 cases of food poisoning from a hotel. Uh, what we do is we are going to collect the food and the vomiters from the patient, or the feces from the patient. So these are the different things what we can collect. We can collect vomiters and the fetus from the patient, whereas the food is always collected from the source. And finally, first we have to go for the direct microscopy for the parasites and the culture test for bacteria and other things. And we should always, apart from this direct bacteria and the parasite, we should always check for the bacterial toxins. As we can remember, as you can remember, the food poisoning, it is usually because depending upon the timing. So if you can say like salmonella and bacillus cereus and vibrio cholera, so apart from depending upon the timings, we can actually divide the pathogens. And apart from that, even the toxins play an important role in causing the food poisoning. So always you have to go for analysis of the food products for the total microbial count and the coliforms. So before talking about the prevention of food contamination, now let us see what is the what are the tests what we can do once we collect the sample. Again, here we have the viable plate method, or what we call it as a standard plate method. It is the standard method followed for the bacteriological examination of food. So this is the one. So food sampling. So here what is it is, what is your source of or what is your sample? Either is it the food material, or you have to check it from the surface of the food. There are two things. If it is a food material, you can take 10 grams of the food and you just mince it in a sterile container with 90 ml of sterile diluent, and you just put it in the agar plates. If at all you are suspecting any contamination of the surface, for example any fruits or the vegetables, such intact vegetable, if you are suspecting any source from the surface of the fruits and vegetables, then what you have to do is you have to take 100 grams of that food, and you have to put it in 100 ml of sterile water, and it should be properly shaken. It should be, you have to shake that particular solution, or the particular mixture, that is with the food and the 100 ml of water, so that all bacteria which is present on its surface should come out and it should get dissolved in the water. So these are the two samples. One is direct food. Take 10 grams of the food and put it in 90 ml of water, or you take 100 grams of the food and you just put 100 ml of sterile water and shake it until the bacteria comes and resides in that sterile water. Then what we have to do, it is a regular process. Then you have to go for the serial dilutions of this diluent and put it on the McConkey broth and look for the growth. So that is what we do. And if there is any growth, then we of any of the bacteria, like how we mentioned, like any staphylococcus or any salmonella, vibrio cholera, or anything, we put it as a source of infection. So how do you prevent the food contamination? The food may be contaminated prior to cooking. So maybe from the infected animals, it can be prevented by, always we know it, proper cooking and allowing the heat to penetrate to the all parts of the food. Then always it should be remembered, refrigeration does not kill the microorganisms. It will just inhibit their growth. So what will happen, you get some products and you just refrigerate it, and you are again reheating it. That means you are providing the best environment for the bacteria to reactivate it. So that is why it always been said like reheated food is a source of food poisoning. I think it should all strike to your brains, like into your minds, like what are the reheated foods. Like if you can remember all the fried rice, gobi, everything what would love to eat, usually those are the ones which are reheated. They prepare it sometime long back, and when you go they'll just reheat it and give it. So if it contains enterotoxins of staphylococcus aureus or anything, so you're going to definitely suffer from the food poisoning. So always you have to maintain the hygiene practices, and water used should be in a good quality. So these are the different ways how you can prevent the food contamination. So this is all about the bacteriology of water, uh, air, milk and food. I hope you have understood. So the most important ones being the bacteriology of water, presumptive method. So it is always the presumptive method, differential coliform count, membrane filtration. Always remember whenever the exam in the exam the questions are given, at least you should write the headings. In case of testing, it is the circle plate method and the slit sampler method. In case of milk, it is methylene blue reduction, methylene blue test, phosphatase test, turbidity test. In case of food, it is just the viable plate method. I hope everyone understood these things. Yeah, I'll take the attendance. Okay, thank you.